Last year, I spoke at this president's lecture about a lot of the research that my lab has been doing over the last 10 or 20 years, actually. This year, I decided to do something a little bit different and focus on something that's completely new, and to be honest, quite unfamiliar territory for me. And potentially it's unfamiliar territory for several people. I imagine there are people in this room who know more about this topic than I do, but it's certainly something that people have been talking about quite a lot in the last year in particular, but certainly over the last few years. And I thought it would be an interesting thing to think about from the perspective of the Linnean Society, from the perspective of people who are fascinated by and really dedicated to the natural world and the study of the natural world. And so that's why today I decided we should talk about artificial intelligence, or AI, and how it is interacting with the natural world and with those of us who study the natural world. So just briefly, for those of you who aren't familiar with the work that we do in my group at the Natural History Museum, what I'm really interested in is trying to understand what intrinsic and extrinsic factors influence the tempo and mode of evolution of organismal form, which is a very fancy way of saying, why do animals look the way that they do? And how have they evolved through deep time? And so in my lab, we bring together lots of different kinds of data and lots of different kinds of information to try to understand what factors, things like developmental mode, diet, other aspects of ecology and habitat, how do these different interactions or these different factors really shape the pace at which animals in particular evolve, how quickly they evolve, and ultimately, why do they look the way they do? And some of the questions we're interested in are things like, why do some forms seem to evolve over and over in different groups, while other things that we can imagine never seem to evolve at all? Now, to answer these kinds of questions, we really rely on big data sets. So we do lots of comparative analyses across thousands of species, actually, and we try to capture their shape in really high resolution. So here is a figure from one of our recent papers that we published about 2 years ago now. And this is a study of mammal skull evolution. Mammals are kind of what I call my home clade. They're what I feel most comfortable with, although actually working on things quite distant from mammals in most days lately. And this was an analysis where we use our understanding of the diversity of forms across mammals, living and extinct, and our understanding of their relationships to try to reconstruct the rate of evolution, and again, how those various factors influence how quickly they've evolved, and how that rate has changed over the course of the last 80 million years, give or take. Now, this kind of data set is really exciting, but it is also incredibly hard to put together. So here's an image of kind of the whole project that this was part of. And in fact, new fellow in the corner is the one in the new-ish fellow in the corner is the one in the video there scanning this dinosaur. And the larger project was trying to look at skull evolution across all of tetrapods. And so for that project, first of all, we had to gather our data. So we traveled around to institutions across the world, including our own, to get 3D scans of these different specimens. And then once we had all those specimens, we had to come up with a way to actually capture their shape. And we really wanted to capture the complexity of these organisms, and not just use a few landmarks or a few length measurements, which is sort of what people had been doing until that point. So we use this approach called semi-landmark analysis, where you essentially place some manual landmarks, these 3D coordinates, onto your objects digitally. And these are the red and yellow dots in that top bird skull, if you can see that. And then we use a semi-automated pipeline that places these high-density landmarks on top of the skull using a template that then morphs the shape of the skull and projects those landmarks onto each object. And that creates this really nice high-dimensional representation of the shape of these organisms. And we can apply this approach across the few thousand specimens in our data set. Now, what we end up with is something pretty spectacular. So this is a representation of some of the data in our data set, again, a few thousand species ultimately. And you can see we're capturing their skull shape in quite a lot of detail compared to if we use traditional approaches. Really, the best we could have done is about a dozen points, give or take, 18 if we really pushed some of our definitions, which really left most of the variation unsampled. If you want to understand the evolution of diversity, you want to understand the evolution of variation across species, it just wasn't capturing what we wanted to capture. So that was really exciting. but it was also really, really hard. And definitely a lot harder than we thought it was going to be at the beginning. Certainly a lot harder than I thought it was when I wrote that grant. And so it actually took a couple of years of putting our heads together before we really cracked the approach and figured out how to make it workable. And even now, that's why whenever I say semi-automated, I feel like that's a real overstatement of the automated part. And so the reality of it is that it's much more like this, right? It's for the Star Wars fans here, it was extremely hard to do, extremely hard to gather the data in the first place, although that ultimately, the scans was the easy part. But coming up with a way to actually capture those 3D representations, or the data that we could actually analyze, was incredibly difficult and takes a lot of time. And so that means as we think about going forward into other groups, and lately I've been working quite a lot on insects and trying to really tackle the diversification of this group that represents the vast majority of known species, right? Over 1 million known species. The biggest analyses that include multivariate traits, so it's essentially looking at something more than body size for insects. Really, anything that's sampling across species tops out at about 400 species, which I think you can all appreciate is a really tiny sample of the diversity of this group. So if we want to try to understand the evolution of phenotype in these groups, we need to find something better than what we have currently available, which are the approaches that my lab developed. And of course, getting these data are becoming easier and easier. So this right here, the little video there, was a robot arm system that's at the Diamond Light Source in Oxfordshire. And using that robot arm, we can scan, and we have been scanning up to 300 insects a day using these high resolution CT scans, which is amazing. 300 insects a day, we can actually start to think about capturing some reasonable representation of their diversity using these approaches. But we have no way of actually pulling anything useful out of these immense scans. And so this is the beginning of a journey for my lab into looking at AI, or artificial intelligence. So this is a little schematic that one of my postdocs, Marco, knocked up yesterday, actually, for a paper that we've just had accepted, looking at the opportunities and challenges in applying AI to evolutionary morphology. And this paper is actually a bit of an experiment in itself. It was trying to bring my team together after COVID and after everybody kind of got used to working in isolation. And for me as a scientist, I find that really I can do the science, but I don't enjoy it if I'm doing it by myself. And I certainly don't do nearly as good of a job at it if I'm not bouncing ideas off of people. And so I really wanted to find a way to bring everybody together. And it was right when people were really starting to talk about AI in a lot of detail. Over the last decade, every now and again, people have said, you should try machine learning. And we'd look at it, and it never quite worked back then. But with all the new advancements, we thought, let's look at this again. And so we set ourselves the challenge of looking at how AI could solve some of our problems. And the result is, I think it's about a 120-page long paper that is in press. And I got fancy this morning and made a QR code if any of you want to see the preprint online. You can go ahead and check that out. And there's a number of approaches or feelings that people have about AI. And maybe most of you are a bit more open to these new things than I sometimes am. But if you look at the evolution of where we are in terms of our comfort with technology and with new approaches, you do get set in your ways, but things have changed very quickly. I quite like this image because actually the second one, the person walking with a phone in their hand, reminds me of the fact when I moved my lab to the Natural History Museum about 7 years ago. I was filming a documentary there just in the first few months I was there. And they wanted me to just walk through the hallway and the main hallway in Hinsey Hall at the museum, just for some background footage. And I was walking and they were like, well, just do something normal. Like look at your phone while you're walking. Cause I was being very awkward walking down the steps. And I thought, I never look at my phone. Well, to that point, I had never walked around looking at my phone. Whereas, today walking through, I nearly ran into a door because I basically walk around like that person constantly. And so you can imagine pretty quickly how we would go from people who are completely tech-free to ones who are using retinal scans for everything, and really kind of locked into this new tech. Now, there's obviously a lot of discussion over where this could lead, right? So there's this kind of concern, right, that robots will replace all of us. Now, as a scientist, I like to think that, there will always be a role in the world for scientists because we generate amazing ideas, and it's just not something that can be replicated. But as David Rawlinson sent me yesterday or 2 days ago, of course, there is now an AI scientist that is able to develop and run its own experiments. So who knows, maybe we are actually closer to this sort of dystopian vision of what AI will do than what we imagine, but I don't think that's the case. But it is true that there is, when you first start diving into AI and what it offers, there's actually quite a barrier. And so part of this, writing this paper, was really getting our heads around what all these terms mean. And so for those of you who don't know all these terms, I've put them up here. Again, if you want to read more about the history of AI, you can look at this book or this paper, because to start with, we kind of went back to what we're comfortable with, which is evolution, right? So we can look at the evolution of these methods and of these terms. And actually, that's very helpful, because what you start to realize is actually a lot of what we've been doing actually falls into the category of AI. A lot of the methods, such as principal components analysis or cluster analyses, actually are types of machine learning. And so that actually gives us something pretty familiar that we can start to think about in terms of what is AI actually doing, right? And it's literally, when you think about machine learning, for example, which is a fairly standard thing, it's really just getting systems to automatically learn, decide, predict, any of these kinds of analyses of data that are pretty standard without being explicitly programmed to do that. Now, the next jump really is to deep learning. And deep learning is essentially a more complex version of machine learning, where the neural networks, which are these interconnected nodes that are processing your analyses, are going through much more complex layers. Now, one thing that actually was, to me, was surprising is computer vision is actually, and again, maybe all of you know this already, but computer vision is actually independent of AI. It overlaps in many ways, but actually is much broader than AI. And so it doesn't actually fit within this entire structure. But there is this kind of natural relationship, a hierarchical relationship across these different kinds of artificial intelligence, where we go from something that has actually been around and we've all been using for decades, such as just basic AI and machine learning, to really the big new thing that has changed everything in the last few years, which is deep learning, where we're actually doing a level of analysis that emulates what the brain does, many layers of neural networks processing our data. Now, of course, all of this is pretty, sounds, very, very autonomous, but of course, these networks are working off of what we put into them, right? So there's different kinds of learning strategies that these different AI networks use. Supervised learning is what actually almost everything that anybody uses actually works off of. And this is where we are creating a training data set using some manual annotations of our data or whatever, and feeding that into the system to learn. And then it learns from that and trains a model, and then it runs that on our new data. Increasingly, there's development of approaches that don't need that much input to actually work. And that's something that's really quite exciting, because actually developing these training data sets is nearly as painful as just doing it yourself. And so there's semi-supervised learning, which uses a mix of labeled and unlabeled data. In this case, I'm using examples of our work on CT scans of various mammal skulls. And then there's also the opportunity to just have really rubbish data. And so one of the things we're playing around with is, if you just put in garbage, do you actually get garbage out, or do you actually get something that's better than garbage? Remains to be seen. I don't know yet. But it's interesting to think about these different ways in which things are progressing to become more autonomous, whereas right now, most of what you use is definitely using kind of supervised learning approaches. Now, I think it's important to think about how quickly things have changed, right? And so this is just pulling publications that use the term artificial intelligence from Web of Science over the last few decades. And you can see that, there was a kind of a steady use of it, probably more in the computer science world for a long time. But, it's really been less than 10 years that it really has started taking off. Now, of course, we're not interested in a lot of those other fields. We are, of course, interested in the natural world. And so what's really interesting is to think about, how has this impacted our field, right? Our studies of the natural world. And so, you can't put the term environment into a search, because obviously computers have environments. But if we look at biodiversity together with AI as search terms, again, doing a simple Web of Science search, you can see that really from about 2020 is when things really, really took off. this field. So that is extremely recent in terms of how much we are using AI to study biodiversity, but also you can see how quickly that rate has increased year on year. And actually what the real difference is because of deep learning. So it's not just, artificial intelligence and machine learning. It is deep learning that has really created this new AI-driven reality for science and actually for really all aspects of our lives. And so you can see that it's, again, really since 2020, but it keeps on going up year on year. Okay, and so I think it's hard to escape headlines about how AI is being used for studying biodiversity, or for ideally helping us understand the biodiversity crisis, and also for figuring out how to solve it. And so here's just a selection of some recent papers and some recent headlines and recent tools that people have been using or developing to apply these new developments in AI, specifically deep learning for the most part, to the topic of biodiversity, and specifically to the aspect of solving the biodiversity crisis. And so you can see that, people are not holding back in terms of what it could possibly do. So how AI can help to save endangered species, and what people are really focusing on are three key areas. One is analyzing vast amounts of data. And going back to the beginning, that is something that immediately piques my interest, since that is something that we do and don't want to do as much as we do. Don't want to gather it anyway ourselves. But people are also using this to set up monitoring systems. And also with the monitoring, then you can start to look at trends through time, both analyzing ones in the past in more detail, but also getting real-time data from various sites. And so again, looking at just some of these examples here, we have monitoring stations at various farms that people are using to actually look at biodiversity. We have accelerating surveys and working with citizen science and community science to get data in place at a far larger scale. We're looking at innovative ways of actually capturing what is out in the environment with things like audio monitoring as opposed to visual monitoring. But of course, we also have things like camera data to actually identify what is walking through a camera trap, or from any other kind of video monitoring system. And so these are all really, really interesting kinds of technology that has really in just a few years transformed our ability to actually track what is out in the wild and actually involve the community much more in the work of protecting biodiversity. So here's a couple of examples I think are really interesting. The fish AI one is one that I've come across in various scientific literature, where they can take images that people are taking on holiday, people upload them to various websites, and you can then monitor what kinds of fish are in different environments. I think you can all appreciate how important that would be. PyTorch Wildlife, again, another kind of similar monitoring approach and helping, again, people with identifying where things are and why. And you can see here an example of just really how this works in many of these cases, where you have trained a data set off of the animals that you expect to see in an area, and then it can go through and identify what those are and tell you with some level of probability what the actual animal is that's going through your field of view. And you can apply this to any kind of organism, and people have, of course, done this. So maybe some of you already use some of these apps when you're out and about, and if so, I think that's great. I think this is going to completely change how we understand our environment. But of course, it's not just about gathering those data, right? People then are using all of these huge amounts of data to actually build better models and make better predictions for how things change. And this is actually also really important, not just from the conservation perspective, but also from the perspective of evolutionary modeling and any other kind of modeling that you do. Because actually, really big data sets are incredibly, and especially things which have complex interactions among many different factors, they can be incredibly hard to run. So for example, that mammal analysis that I showed you at the very beginning from our own work, I mean, that is something that would run on a cluster for about a month before we would get an answer, right? This is an immense amount of time. And that is a relatively tiny data set compared to the amounts of data sets that we're getting now from citizen science. So you need to find better ways of actually analyzing these data, not just gathering them. And so this is an interesting paper that came out a few years ago that actually uses deep learning to analyze a series of different factors, starting from species distributions, but also looking at a variety of factors. So you can see here they have species richness, population density, disturbance, climate change, and phylogenetic diversity as well, which is something that I think we talk about quite a lot in terms of how to prioritize conservation areas. And then feeding this into a complex model and actually bringing in information about policies to inform on where the priority areas should be. And what I think is actually really clever about this is it also takes into account budgets to say, okay, if you have a limited budget, what is the best that you can do for that money to conserve the largest number of species or the most important species? And so you can see how this has real world value very quickly. Again, from something that has really only been around in the scientific literature for the last few years, we're already seeing this kind of impact. Now, I work at the Natural History Museum, as you heard, and of course, there's lots of species and lots of life out wandering around today, but there is a lot of it sitting in museum collections, right? So just, you know, at the Natural History Museum, we have what we say are about 80 million specimens. It's probably closer to 100, actually. There's a couple of people who work there in the room, so you can disagree, but I think if we actually counted everything that's sitting in a bag with a bunch of other specimens, we'd be topping out 100 million. And so that is a lot of data. And in fact, I think it was just last year, there was a paper published that took 73 major natural history collections around the world and tried to quantify how many specimens are in those collections. And it was about 1.1 billion objects that were in those different collections. Now, I think you can all appreciate this is an incredible resource for understanding biodiversity change through time. I was going through one data set of deep sea muds because I have gone really far from mammals somehow just last week, and we have thousands of rows of data for deep sea muds going back in some cases to the 1840s, right? And that's a huge resource for being able to track biodiversity change through time. In that case, we were just looking at the North Atlantic, right? Thinking about the Greenland ice sheet. And so that's a huge resource. Those have not been looked at basically since they've been collected, right? Because it is incredibly hard to process that kind of data. And that is the problem that all of us who work at these museums really struggle with, is how do we actually use these data in critical and useful ways, not just for the kind of, fundamental questions about how the evolution of life, as I'm interested in, but also for how we can track recent biodiversity change. Okay, and so, I mean, this is just for the UK now, where we have, I think it's 137 million specimens across 83 institutions, as shown on this map. And recently, there's been this project that was funded with, I think, about 150 million. Somebody can correct me, but I think it was about 150 million pounds that was funded to digitize these collections and create a database of all the specimens in the UK collections, which will be an immense resource because actually a very small proportion of those collections are even captured in a digital record, right? Even the base data about the specimens is not accessible online, which means we have a huge amount of information about the history of life, deep time to shallow time, that is just not accessible unless you're going to go through cabinets, which, is fine if you want things to happen at the pace they've been happening for the last, couple hundred years. But I think we can all appreciate that we are quickly running out of time to find solutions to this crisis. And the more data we have to inform our actions, the better chance we're going to have to actually come out of this on the other side. Right. And so this program that was just funded, Disco UK, is trying to solve this problem by digitizing essentially the entirety of the UK collection as an open resource for people to then study these data. But again, we can go back to the question of how do you actually then do anything with it? So here's an example of the sorts of AI tools that people are already applying as they're digitizing these specimens. These are examples from the NHM. Part of it is actually just going through and being able to create a digital record of every specimen at scale by capturing both a 2D image of the specimens, but also their labels, because actually transcribing their labels is an incredibly time-consuming step. But if you're going to do anything with a specimen, you need to know where it came from and what it is, right? And when it came from, for that matter. And so they're using a lot of interesting AI tools to actually speed up this process and go from a state where over the last decade or so, the museum has been able to digitize, I think, around 6 million specimens, which is pretty good, but it's, obviously probably 6 to 10%, say, of our collections. And so if that's our pace, we're going to take a couple hundred years to get through it all, right? But now we can speed this up. So part of it is just being able to figure out what we have, but also part of it is figuring out what they actually are. And so this lower example, these beetles here, is actually some work that a PhD student has been doing to actually classify specimens. Because remember that a lot of these species are also not named or not named correctly. And these names have, of course, changed a lot. If anybody, for all of you who work with specimen data, you know the number of times that you think that you've digitized one species and you find out later that's completely something different, or that the name has changed 10 times since the label was written. So again, this takes us back to the problem of what do we do though? We have these great 3D images, say 2D or 3D images of these specimens, and we can now gather them at this incredible rate using AI and robots. What can we do with them? And this kind of brings us back to kind of my interest in AI and why I've bothered actually putting the effort into to learn this instead of the many other things I also want to learn, but just don't have time for. And so there are a lot of tools that people have been developing over the last few years to do some of the base things that we do when we first get new specimens, right? Trying to figure out what they are. And so here again is the example from that PhD student's project where they were able to develop an algorithm to classify these species with new precision. And one thing that I think is really, really interesting about this is that not only could they say, these are species that we already know because we trained our model. This is what they were actually able to also do is identify here are species that don't fit our models. And that's actually something that's not so trivial, right? Because when you think about what AI does, it just works off of what we put in it, right? As I said, you put in garbage, you get out garbage, you put in brilliance, you might get out probably still garbage, but well, something in between, at least something closer to brilliance. But whether it can do anything novel, whether it can tell you something that you don't already know, or that you couldn't figure out if you had enough time, is unknown. But the fact that this is actually able to identify new species is a really, really interesting thing. Now you can imagine for huge portions of the world, right? We by some estimates, we know in tiny fraction of living species. So if you can figure out a way to quickly identify what new species are out in the field from visual data or audio data or anything, that is an incredibly powerful tool. So I thought this was a really exciting new development. And then of course, people are also figuring out how we can use this to do some of the other things that we really need in order to do big analyses, like understanding the relationships among species. And this is a critical piece of any evolutionary analysis, quite frankly. It's also really important for prioritizing conservation, right? As we said, phylogenetic diversity, whether you're capturing something that has a really long and unique evolutionary lineage is a really important thing to consider. But if you don't know how something is related to anything else, you have nowhere to go. And so this was a new paper that came out in January of this year. And actually, this is my first example of where preprints are actually very useful because we put our AI paper up on a preprint server and somebody read it and wrote back and said, hey, great paper. We just had this paper come out, which is really relevant. You should take a look. And it was, of course, incredibly relevant and really interesting, where they basically take existing analyses of evolutionary relationships. They took the traits that were really supporting the phylogenetic tree that was the output of that analysis, and then they fed that in to a new analysis along with a bunch of new image data, and then use that to actually put these new species into a phylogenetic tree at a far more rapid pace than we would normally be able to do. And so that again starts to really speed up the process by which we can actually build a phylogenetic tree. And for again, any of you who have tried to build a phylogenetic tree, it is painful and it can definitely hold back projects for years as you try to figure out how things are related so you can run all your other analyses. So I thought that was incredibly exciting. Now, again, my interest really is about phenotype, trying to understand why things look the way that they do. And there have been a lot of interesting developments in the last few years on this area. So here's a study done by one of my postdocs during his PhD, actually looking at color evolution in bird wings and bird feathers. And this is a project where, and what's interesting about this is both a community science project where they had a lot of, I think, hundreds of individuals going online and actually capturing the area of the feathers from these 2D images. So then they could scan just those areas to see the color and the UV of radiation of the coloration of the feathers. And it took him about the same amount of time as it took for the entire community to do it manually. By the time he actually got the model running, it was about equal time. But then when he ran the model, he could run it in about a day. And he could do in a day what it took, hundreds of people 2 years to do, right? And so it's not, it's not magic. It still takes some time. But when you get it working, it's actually pretty amazing, because then that can be applied to a whole other set of data. So this analysis ultimately was able to analyze about 5,000 bird species, and again, about a day's time. And then they can start to look at the distribution of these traits across a really massive scale. And again, these are the kinds of analyses that just aren't possible until a few years ago. And so this is something that we've been kind of developing in my lab in the last few years. Really, our interest is a field that I would call phenomics, which is where you're trying to really look at phenotype, but in a really high dimensional and organism-wide way, as opposed to just looking at individual traits or just a few traits. And with it's a very difficult thing to do, as I've said, but with AI, you could potentially start to actually tackle big questions. So call it phenomics, which maybe nobody understands that term. But the first thing that we need to do is figure out how to process massive amounts of data. So as I said, we can gather these huge volumes of CT scans really rapidly now. But CT segmentations, which is where you actually take your stack of X-ray images and actually pull out what you're interested in, takes a really long time. For a tricky specimen, it can take days. For an easy specimen, half an hour. But for a tricky specimen, it can take days actually to segment out your 3D scan. But through using AI approaches, deep learning in particular, we've actually managed to get this down to a really, really rapid level. And so one of the students in my lab, James, he has been working on forams. Again, I have really gone very far from mammals lately. But what he was able to show is that it was just a very small training data set, even just a few specimens that he did manually. So that would take, a day or two to do manually. He could actually train a model that could then process all of his scans with a really high level of fidelity to the original data. And he did this by comparing a manually annotated data set versus an AI one. And you can see the fit of the line is extremely good. Now, what this means then is he can take this immense data set of CT scans of foraminifera and start to look at really interesting things. So as I said, normally it would take, probably months to process that level of data. Actually, did I go back? Oh, here it is, sorry, skipped one. He was actually able to process 1,700 specimens in about 2 to 3 days, was a conservative estimate of how quickly he could get through it. It was probably a little bit shorter than that. And with those data now, he has this incredibly refined picture of how these different species of forams have changed through the Pliocene, essentially through this kind of massive shift in global climates. And what he's trying to understand then is what are the cryptic morphological changes that happen through speciation? And that's really difficult to do. And he's trying to really bridge microevolution with macroevolution, which has been really tricky because sample size has always been the constraint. And now with these approaches, all of a sudden, sample size is not a constraint. He literally took about a week to pull all these phenotypic data from 1,700 specimens and have his analyses done. And then when I think back to the fact that, my mammal skull data set, quite frankly, took about 7 years to get 300 specimens. He also ran my mammal data set through his pipeline, and it took about a day. It's totally cool. I did not cry myself to sleep at night. But you can do really, really amazing things now, looking at the moment of divergence between species, essentially, and how that actually affects their variation, how that relates to these large-scale shifts in global climate, and how that changes the distribution of their morphologies at this incredibly fine scale. And so we have various approaches that we can apply to these data sets, essentially. I think this was meant to be the next slide, but this is another approach that people have used to pull essentially 3D landmarks, or in this case, 2D landmarks off of bryozoans, I think in this example, using this automated approach. And again, this is the thing that we were doing at the very beginning that was incredibly painful in those early slides, that took an immense amount of time, hours per specimen. Quite frankly, that's how long it would take us to put our landmarks down. But there are AI algorithms that can get pretty close, at least for 2D and increasingly, I think, for 3D specimens. And so we can start to look at a world where, just a few years ago, literally 2 to 3 years ago, what we would be doing is getting our 3D scans, we'd be manually segmenting them, then we would have to, pull out our different parts of the skull using our landmarks or whatever. And then we could actually get some 3D landmarks that we could analyze. You can see the calendar there shows that this process takes months and years, quite frankly. But now we can do most of this through an automated process that takes seconds, which is just amazing because it literally is the last 2 years that this has become possible, maybe even just the last 1 year. Now, that's not to say that every part of this is amazing. And I want to then now talk a little bit about where it isn't helpful or where it hasn't actually panned out, because it isn't like a cure-all for every issue. And one thing that actually we've been trying to do is take these scans and pull out the things that we're actually interested in. How do we actually parcellate them or fragment them into different parts of the morphology? And there are various tools out there that we've tried with our various data sets, and they quite frankly didn't work. And we tried, we spent about 1.5 years actually on one project trying to train models that could actually do this reliably. And the idea, the plan, right, is that we could do all of this through AI, acquire huge vast amounts of data through these robot pipelines, use deep learning to separate out the different aspects that we wanted, apply our landmark-free approach, which does work amazingly well, and then we can actually start to analyze huge numbers of various species. So in this case, this is our pilot data set of insects, and even with a slightly clunky version of this, we were able to analyze what is already essentially the largest cross-insect multivariate analysis that's been conducted. But that glosses over a lot of issues actually in this pipeline. And so I think the best demonstration of how this hasn't worked is a different study that we're doing, looking at mammal skulls again, going back to my home clade, where we're trying to understand not just overall shape, but actually the evolution of these really minor features, the sutures. These are the joints between your cranial bones. You can probably potentially feel some of them. You can certainly feel them on children. And we wanted to look at the evolution of these sutures, which can either be very simple, as in the myocastor, or they can be incredibly complex in 3 dimensions. And so what we wanted to do is take our CT scans, train an AI model to pull out the sutures, which are essentially the gaps between the bones, right? And so they actually look quite a lot like the background, which is difficult. And then magically be able to quantify all of this. And we tried this again for about 1.5 years. And the best that we could get to was about this level, which is pretty good. You can see the sutures in there, but you can also see a lot of other stuff. And we've tried many, many different new AI techniques to try and get to this. Before we thought, let's go back a step to something a little bit more traditional. And we just went back to kind of basic computer vision and thresholding. I'm sorry, that's what a 3D suture looks like when you pull it out of one of these models. It's pretty good, but there's a lot of garbage in there. And actually, so what we've done in this case is just go back to old computer vision, but with a bit of fancier code to essentially pull out these sutures. Now, hopefully you can see that this is quite beautiful. You can take this skull and really in a matter of, say, I mean, the way it runs probably takes about half an hour, but actual individual time, it's a lot faster. You can pull out this really beautiful 3D map of all the skull sutures. And this is, again, the kind of data set that you just don't see any comparative analyses of, because actually doing it manually takes so long. So yeah, there's, say, in the entire history of people looking at sutures, which is important for all kinds of, developmental and functional aspects, and obviously is linked to a lot of developmental disorders in humans. The biggest comparative analyses would be a couple of species, because it is so hard to pull out those data, and usually just looking at the kind of surface as opposed to what's going on under the surface. But now we can do this literally in minutes, and we pulled out just, I think, in the last, well, actually yesterday I set them the task of saying, how many can you do in one day, get through it? And, in a single day, you can certainly pull out, I would say 20 or so specimens potentially. So, we can pull out hundreds of different species in a matter of weeks, whereas normally that would be an entire lifetime's effort, quite frankly. But this is a case where AI actually let us down. We had to go back to normal approaches. And so AI doesn't always work. This approach also works very beautifully on our 4AM data set. So it's just a lovely image of looking at all the different chambers and where we can draw on developmental data alongside deep time data to really bridge these different scales. Now, here's another thing that AI can't do. Can't go out into the field. Well, maybe with camera traps and things like that, it can. But it can't dig, I don't think, until they have robots, I guess, that they control directly. But one thing that will never cease to be necessary is for people to go out and discover things, right? And go out and look for new data. And this is something that I think, and I think probably many of you will agree, is really overlooked when we talk about exciting new technology, right? We talk about everything that we have in our collections, which is amazing. And we talk about how rapidly we can gather those data from the collections, which is absolutely amazing and really central to my entire research program. But what we can't do is fill gaps, right? And when we have previously looked at the distribution of specimens in collections, certainly at various points in the past, you can see that there's huge areas of the world that are basically unexplored. And so until we get out there and actually continue to find these species and continue to search in underexplored areas, then this will never change. And so this is one thing that is really important to always remember. AI is only going to give you what you've already put into it, right? Sure, it can maybe identify new species once you've trained it off of a species but it can't go out and look for new things. And so we will always need to get our boots dirty to discover new species. And then really the last thing that I really want to touch on is something that I think a lot of people don't think about enough, which is this stuff is expensive. And I don't mean expensive in terms of money, and it's expensive in terms of money too, to be honest, but it is incredibly expensive in terms of energy usage. And so AI in terms of, these are actually a swap, in terms of energy usage is far, far more expensive than running a normal computer program. And so, there's been a lot of comparisons, for example, of how much more energy in water also it takes if you ask a ChatGPT a question as opposed to just a normal Google search. And you can see here that the emissions of some of the major producers or users of AI, like Google and ChatGPT, are hugely increasing their energy use at a time when all of them have made commitments to reducing it, right? And they've all started to acknowledge that this is a huge problem. If they're going to change all their search algorithms over to using AI, we are never going to hit the goals that we all know we absolutely have to hit, right? And so there's some really, really shocking numbers in here. So you can see in the caption to the figure about ChatGPT that energy consumption by artificial intelligence is predicted to consume twice as much energy as the whole of France by 2030. There's another one in here, maybe it's on this slide, about the amount of water that is being used by AI, and that essentially, it's half the amount of the UK by 2027 is the amount of water that's being used to run these kind of common algorithms that we all think is really fun to like make some weird figure, or, I used it this morning actually to do a translation into Hindi. Don't tell my mom, I can't actually do it myself. But every time you do that, it's using an immense amount. And so people need to think about why they're using it and use it carefully. You can't just throw everything in there and forget the incredible environmental footprint of these new technologies. And so what we do, at least in our lab, is that we test things out on small data sets before we run the big models to make sure we're not wasting energy by running things that have no purpose, that are not going to work. So we do that to try and be more responsible about our use of these technologies. There are a lot of guidelines that people have put out there about how you can use AI. Obviously, we want to be able to take advantage of this new technology. As I've said throughout this talk, it gives us immense potential to do things that we just cannot do scientifically. It gives us immense potential to actually really get the critical data that we need to solve the current biodiversity crisis and make better predictions. But if we're actually using more water and more electricity to do that, then actually maybe it's not really worth it. And so we need to make sure that we're thinking about how we're using it and use it appropriately and efficiently and effectively when we do apply these approaches. Okay, so there's that point there. This is, I think, the thing I was pointing to. So a single ChatGPT query consumes 15 times more energy than a simple Google search of the same thing. So don't use ChatGPT if you could just use Google search. Although, of course, Google search is increasingly using AI. So I just want everybody to keep that in mind. It is not free. And the other thing that I think is really important to think about with AI is that a lot of it is a black box, right? And the real push in recent years has been towards open data and open science, right? Giving people full access to your data, making your code available so that people can replicate your analyses. But a lot of the models that are being built today and being run by some of the big companies that are using the data that we are all putting out there actually doesn't make the models available. And that actually means that we don't actually know the accuracy, we can't replicate those results. And so I think a real worry also is that AI will continue to close in information and make it less accessible when we should all be moving in the other direction of having that information more freely available. It also puts all the information and, quite frankly, the power in the hands of a few, whereas hopefully we can continue to democratize data, democratize science. Okay, so I'll stop there so we can have a bit of a chat. But I just want to thank my lab. The lab at the top are the ones that had to do everything manually, and this very relaxed group down here used a lot more AI. So you can see it has made a huge difference. Also all of my collaborators, and especially all the people around the world who give us access to their collections, without which we could not do any of the work that we do. And obviously also all the people that fund our research. With that, I'm happy to talk more about this. I'll figure about some of the things that we're doing with our AI. And again, there's a QR code if you want to look up our paper. So thank you all, and I'm happy to open it up to questions now.